Welcome to this evening talk. I'm happy that so many people found their way here. This is going to be a very informal, or at least informal, discussion about gene technology and societal and ethical aspects of that technology. We have a panel here with a relatively diverse background, but they all have one thing in common, and that's that they have some familiarity and some experience with working with gene editing or gene technology. My name is Anders Braarud Hanssen. I am the current center manager for Digital Life Norway. I'm also the RRI coordinator, which is an acronym for Responsible Research and Innovation. And if you look at me touching my face all the time, I realized that I had to change my glasses. So it's falling off my face constantly, so I apologize. And if I start to read something that's not right, it's because I'm just kind of, swapping back and forth between focus and non-focus. That's just a little caveat. Anyway, so I'll have a brief introduction. I'll start with Professor Šikšnys. Of course, most of you have either been to his lecture or have heard of him before, but very briefly, he is a professor of biochemistry at Vilnius University, and he's also chief scientist at Vilnius University Institute of Biotechnology. Thank you for being here. And also, Trygve Brautaset, you are a professor of synthetic biology at NTNU, and your research focus is within microbial molecular biology. Is that right? Mm-hmm. You're also the now current leader of the Biotechnology Advisory Board. Is that right?
Deputy leader.
Deputy leader,
yes. Yep. To make it correct.
Make it correct. It's very important. And also currently a member of the Gene Technology Board.
Yeah, so that's Genteknologiutvalg. I don't know what that is in English, but that has been passed. So we delivered our report in June this year.
Yeah.
So that has been finished. Yes.
Interesting. So quite relevant.
Yes, very relevant.
And also Marianne Fyhn, professor of neuroscience at the University of Oslo. And you have been working with animal models and CRISPR, is that correct?
Yeah.
And your work is primarily focusing on the hippocampus and spatial memory and navigation, is that right?
Yeah, among other things. Yeah.
I got it right. Fantastic. So we'll talk a little bit about your familiarity with CRISPR as well. And finally, Dorothy Dankel, you're a senior scientist at SINTEF in Bergen. Your main research focus is marine science, and you're working also in the area of RRI, Responsible Research and Innovation.
Correct.
So yeah, great diverse backgrounds, but I'm excited to delve into the societal, ethical, moral questions around this topic of gene editing and CRISPR. And I'll start with Trygve. Maybe we can kind of unpack at least the lower hanging fruit when it comes to this. It's relatively complex, and it's not so easy to kind of dissect what's an ethical question and what's related to scientific uncertainty. So could you say something about the Biotechnology Advisory Board's kind of sifting around the balancing between scientific uncertainty and the risks versus the benefits and the opportunities and kind of moral obligation to do something with the technology?
Yeah.
Brief.
So first, when we talk about CRISPR-Cas, which we are doing, I feel, so you have men without hats, and then you have men with hats. So today I have kind of three different hats. Yeah. One is the Biotech Advisory Board, and one is the Gene Technology Utvalg, and also as a researcher using this technology myself. You asked about the Biotechnology Advisory Board. So this report, we haven't taken any, we haven't made our opinions about it, so I shouldn't say so much about it in that respect. But you heard Håvard today talk about a report that we sent on sustainability in food sector, where I think that which has been sent, so that is fine, which I think was kind of a positive attitude to taking use of GMO as long as it is done in a good purpose. So that is at least the view that we have now before we look into this report. What I could say, I work with microorganisms, and you heard the lecture this morning. These fantastic systems were discovered in microorganisms, but like what I think is interesting in a way is that so you made a discovery of a defense system, and then it was turned into technology for genome editing. And for us working with microorganisms, this has not been such a big revolution in gene technology as they have it in higher animals, even in human cells, in higher organisms. And the simple reason is that we had genetic tools to do these things before CRISPR came, like homologous recombination. But still, bacteria are different, and if you work with more sophisticated bacteria, then the tools that have been developed over the years for the typical model organisms, they don't work there. So, for example, in my case, we have a bacterium where we could not at all use established techniques, and then CRISPR-Cas was really the breakthrough for us to make knockouts on the chromosome. And ethics, I mean, when you work with bacteria, ethics is relatively much simpler. I mean, we don't know scientifically if they have a soul, if they can be sad or happy, or if they can feel pain, but we don't believe that they can. So, and most of this is also meant for contained environment. So ethical, I know that people working with animals and things like that, it's much of a bigger issue than it is for us.
Yeah. Do you mind if I just butt in? Because I would like to unpack that a little bit, Trygve. Because, yeah, you're thinking maybe about the ethics right then and there. But with RRI, we try to think also anticipate the knowledge that I'm making here and proving that can be done in this organism can be maybe used in a mammal or in a human. And so the anticipatory ethics is an important dimension. So you might not think it's so important, but it could really be.
Yeah, see your point.
I just had to make that point before it was
Yeah, and I mean, talking about anticipation, you know, I listened to your lecture today, Professor Šikšnys, and I was wondering, I mean, even though you're working with a science that's here and now, you're kind of peering into the future. And the one question I had when I was listening to your talk was like, do you grapple with any kind of ethical or kind of, you know, like, are there like fundamental uncertainties that make you think about ethical questions while you're doing your research?
You know, I agree with Trygve. When you work with bacteria, you do not think about ethics at the very beginning. Yeah, so because what you are doing, you are trying to address very basic biological questions, how the system works, what's doing in bacteria. So then, actually, there is, of course, the moment of discovery. So when you recognize that, okay, you discovered something that behaves differently, and Trygve said that before there were no tools to do genome editing in mammals. No, there were tools, actually. There were talons and there were zinc fingers. But actually, these were tools that were available only for a bunch of people, because these tools were really difficult to reprogram. But actually with CRISPR, though the reprogramming became really very simple, you just change RNA and you can direct it to any target in the genome. And then this made genome editing kind of available for every student. So that kind of democratized the genome editing field. And then, of course, when this system was transferred to mammals, so then actually these ethical questions arrived. But yes. So.
But can I just follow up then? Because, okay, I've tried to do my homework, but I understand that I have the understanding of this scientific area like a Labrador. Like there's no way I can get my head around it. But okay, and you have to correct me if I get the question wrong, please. So, you know, when it comes to uncertainties, I've understood when it comes to using the Cas9 protein, for example, it's a protein, right?
Yeah.
Yes.
Keep going.
There is. I'll try to get this question, formulated. So, from what I understood, there are so-called off-target effects.
Yes, there are.
And there's a fundamental uncertainty when it comes to off-target effects, right? You can't really anticipate them a lot. Is that correct?
Yes, so this is a logical outcome, these off-target effects. So before I was working on restriction enzymes, and some people in the audience who tried to do some cuts of DNA using restriction enzyme, they know that they actually have to follow the manufacturer's instructions, because if you add too much restriction enzyme, it will start cutting somewhere else. Yeah. So, but it's kind of not big difference with CRISPR and Cas9. So these off-targets could happen, and that's kind of, you have to deal with that. Yeah. So you know that off-targets happen, and then the next question is how to avoid them. And yeah, so.
I see. So, okay, I'll come back to when it comes to, like, the potential of using this in human clinical trials, for example. But I'll go on and start with a question related to what Dorothy is doing, because you've been working with salmon and sterile salmon, is that right? So you've been working more about, can you describe a little bit about the RI aspect of your work related to CRISPR salmon?
Yeah, so it actually started in January 2013 with this project that was the precursor to the Center for Digital Life. What was it called again? Bioteknologi. Or something like that. And the salmon sterile project, the idea was that for Norwegian salmon, which is a big major export and a big moneymaker for Norway, escapees are a big problem, one of the big bottlenecks to increasing production besides different environmental effects. But the interbreeding with wild salmon is a big threat. And we know that escapees happen. So the idea was, could we sterilize the farm salmon? The first idea was in a vaccine approach, because that's a commonly used approach, and that would be well accepted by society. But then Anna Vargelius, a researcher at the Institute of Marine Research, she brought up in the kickoff meeting this new paper that had CRISPR gene editing, because that, of course, came out around September of the previous year. And she said, I want to try this. I want to see if we can get a salmon egg to be sterile. Try this CRISPR thing. And the industry members said, is this GMO? Because if this is GMO, we don't want this. We cannot associate Norwegian salmon with GMO. It ruins the whole made in Norway pure salmon brand that they've built up. And she's like, no, I mean, we would just try it to see if it worked. The basic science is discovery. See if it works in Atlantic salmon. And right after that, when they said, yeah, you can try, we'll figure it out. Right after that, we went to Boston. My husband was at Harvard for a year, and I was nursing. I was on maternal leave with our second daughter. And we get a knock on the door, and it was Malina Klausnitzer, and she was working with Simon. And she said, the CRISPR works, the CRISPR works, the CRISPR works. This is June 2013. And I'm sitting here nursing, and I have all these maternal hormones, and I'm like, CRISPR? I think I heard something about that before. And she's like, it works. She was at the lab at Broad and MIT and Harvard, and she had done this first experiment where for the FTO locus for obesity was known in humans, and she knocked out a single nucleotide in a mouse and then reversed engineered it. She reversed and got the obese mouse, and then made the obese mouse, another mouse, skinny. So it was like a beautiful example of how CRISPR could work, and that application to human disease. And it was when I got back then to Norway, and Anna Vargelius had started the CRISPR experiments, I was like, I am going to document this with an RRI frame, because it was the first time that it was used for Atlantic salmon. Atlantic salmon is a big moneymaker in Norway. Where is this going to go? It was very interesting from an RRI perspective to use social science, use RRI, and write that context. That it's a kind of a historical ethnography, if you will, of what was happening. Yeah. 10 years later, Anna Vargelius is leading the report committee, which is a huge commission of trust, considering she has really dedicated her career in developing this. The first time they used it on salmon, it worked we had a sterile salmon. That paper was published in 2014, and now those original salmon are big now in the lab at Malta, and now we have many other follow-up projects, and it's been really fascinating to follow that trajectory.
I think it's, just for full disclosure, the first time I encountered the word CRISPR was actually because you invited me to a workshop. That's what it was in 2017 or something. Yeah. And I was, listening to your colleagues at Havforskningsinstituttet, I can't remember.
Institute of Marine Research.
Institute of Marine Research. And the way they talked about their science, you can see their excitement.
Oh yeah.
Because they were like, they talked about the anticipation of going up to this big tank and then wait for the results. Have they been, they did work and they opened the hatch. And they were so excited. And I was like, wow, you're so lucky to be part of something so groundbreaking. As it was unfolding.
Because it's a big difference, this working with the bacterium in the lab, in the basic science, but then it is basic and applied at the same time. And to have also see how the industry reacts to that. In the beginning when they say, nope, if it's GMO, we're not going to be associated with it. And actually, truth be told, I was not, there was one particular industry member, when I came back from maternity leave, and I said, Dorothy, what are you going to do for your work package? I'm like, I'm going to write about CRISPR. And the industry member said, no, you're not. We do not want a publication coming out where you're going to scare everyone and say that now we're going to use gene editing in salmon. That is not part of this, the communication plan of this project. And those were legitimate fears that the public doesn't know what's going on. Their public, even today, I think, are very ignorant of what this is and what it could be, and having a debate about what should be.
Yeah, and that's, it's not only about, of course, animal ethics, but it brings me to my question to Marianne, of course, because you've been working with animals, animal models using CRISPR. Can you just very briefly describe what kind of research that is and what kind of application that has for neuroscience? And also if you have been kind of contemplating the ethical implications of it with regards to animal ethics.
Sure. So I work with neuroscience, and we use rodents as an animal model. And I think that there are two aspects of it. So one is that we now have available tools to apply to the brain, which is extremely complex. So we need kind of tools that are targeted and precise in order to interfere and intervene with these neural circuitries, and then record the behavioral effects or the effects of the neural circuitry. So then the CRISPR allows us to do that, which was not possible before. And one difference is that we work with somatic cells. That means that these are cells that they don't divide anymore. So they are adult cells. So that means that now we have a tool where we could intervene with the adult cells. We are not dependent on kind of germ cells. So I think that in that regard, it's good for the animal welfare, because we actually use less animals with these techniques, because they are more precise and more targeted compared to what we had to do with before, transgenic mice. We had to breed many different generations. We only selected the ones that we wanted and then sacrificed all of the rest. So that was very time-consuming. It cost a lot of money, and also many animal lives were then sacrificed. Yeah. So I think in that regard, it has kind of opened a new era for improved animal welfare. And then on the other side, what with the many people in neuroscience, including me, we are very interested in the basic science, but much of the focus is also to try to understand what goes wrong in disease. And then many of these neurological and brain diseases, we don't understand them. We don't have a cure, meaning we don't have any medication or treatments for them. And so then this complex brain with these diseases that we don't understand, then we need some more high throughput ways of trying to dissect what goes wrong. And there also CRISPR could help us, because as you say, you could just design a new RNA, and you could have many of these guides, and then you could have them combined, which is actually what happens in the brain. There's very rarely one single mutation. There's maybe subtle variations to many genes. So I think that's kind of a huge advantage for like the basic and preclinical work. But then when it comes to bringing this to the more to medicine, then it's a completely different game, because then you run into the off-targets effect. You can't allow them. Like if you want to infuse something into my brain, I don't want them to have any off-targets effect. It needs to be very, very safe. So there, and also then, although we could have some discoveries in these diseases, it's a long way from our animal models to what actually happens in the human brain. So there is a big gap, but likely we will hopefully come there. Some other diseases, not including the brain, there's also being used, like CRISPR is used in, say, like, for different blood cell diseases. It's already in use. So it will likely come, and there are some promising diseases where we think it will come. So I think that these discussions for the implications of what we could do, what may be the implications, we need to have them before it's possible to do it, because the research will just, that's a kind of a train that will just go forward.
But you know, as a kind of a like a general question, and you're free to kind of grab that if you want. Of course, if you say we need to have these discussions now, you know, I think there are quite a lot of scientists working in this area that are reluctant to be too speculative, right? Like with regards to the use in humans, this kind of, you know, balancing act between, oh, this is kind of clinical trials, it's about solving medical problems, versus where does the boundary go towards human enhancement, for example, or really going deep into the, you know, the difficult ethical landscape of where you actually are invoking a change that's going to enhance something versus just solving a medical issue or solving a disease or addressing a disease. So, I mean, of course, these discussions are necessary, as you say, but do you think that you guys, you know, as scientists, are willing to go into the deep end, or do you really want to feel like you're keeping it safe and close to your data, and you want to just stay close to what you know instead of trying to anticipate future impacts, implications of what you're doing?
No, I think that, like, for, say, my core basic research interests, then I'm not that much of in a dialogue with the kind of the public in that regard. But when working with these, say, the diseases, you also collaborate with clinicians and also patient organizations. You immediately run into these issues, even though we work with animal models, and everyone knows that it's far away from kind of the clinic or the bedside. So I think that scientists are more and more open and acknowledge that we need to take these discussions. So I think that it's not true that scientists are not interested in those kinds of discussions.
All right.
Yeah, no, I agree, and also I think that sometimes there is like a big step in technology development, like CRISPR-Cas was, that was pushing our capabilities of engineering rapidly forward. And when we listen to your lecture today, that might happen again, because you are still looking for new potential systems, and we don't know what we will find. And it's a little bit analogous to what is happening in artificial intelligence now, which is also used for biochemistry and engineering. We have this AlphaFold program. It's still not perfect, but you can have an imagination. If it can predict 100%, then you can also start doing things the other way around to completely design enzymes from scratch. You don't need to go to the metagenomics or to the genomes, which can accelerate our capabilities dramatically. So I think this is important. So we can have tools that are much faster, for example. I mean, we that use CRISPR, it's not like this. It's a lot of laboratory work to make a mutant. So it's not like a 1 hour work. Suddenly there can come up something that is even more efficient. So yeah, I think it is important to think about these things.
And just to follow up, I think that it's very important to engage in the discussions with general public regarding novel genome editing technologies, because I think that one of the reasons why GMO was rejected by society was because from the very beginning, there were not much engagement with the public. And then technology went to big companies, and then people were making money from that. And that's kind of get a negative just background for GMO technologies. But as we had today, actually, this could be useful, and this could solve very, very many problems that we encounter right now. Yeah, so that's...
I would say there's no doubt that the GMO flop in Europe was in the back of the minds of in the European Commission to get RRI up and going several years ago, because we don't want to spend public money on a science trajectory that's ultimately rejected by the public. I mean, it's a total waste. And the US has had a totally different approach. You know, the ethical, legal, social aspects, ELSA, that really originated in the United States when they saw the potential of genome editing 20 years ago. And then they said, we have to get ethicists involved, because they were, you know, in the US, you always have this kind of the threat of litigation, threat of you want to cover your, make sure you're doing everything really well because you don't want to go to court. Whereas in Europe, there's not a tradition for that. So we see, you know, RRI had a big prominency and in the, you know, biotech calls in the EU and whatnot. And it's really expected now that RRI, and as the Center for Digital Life, has it embedded in. We do not have enough competency with doctors and on RRI issues, or with molecular biologists on RRI. It really should be I ryggmargen, what do you say, in the spine of every scientist, that this understanding what are the potential positives and negatives, how could my science be misused? What is my responsibility in communicating and engaging? How can I engage, not just to inform the public of what I'm doing, but to get feedback and input for the public on dimensions I never even thought of that actually makes better science. And if we talk about, you know, we talk about sustainability and how important that is, we're talking about social political sustainability of molecular biology, that we want that feedback and we want the public to be able to weigh in. They need a certain amount of knowledge. So it's a movement towards, you know, essentially better science.
But as you say that, I was thinking about your comment, Professor Šikšnys, because I was thinking like, okay, would you say then that if the scientists quite early started a deliberation with the public or with other areas of academia and other stakeholders, instead of waiting until the industry kind of, you know, tapped in, that would be a better approach?
Yeah, so I think that it could be a better approach. Yeah, because you start to talk with people and then trying to show people that this thing could be useful rather than deny it. Yeah, so that's kind of.
Yeah. So would you say, and I recall my conversation with Marianne in the break, you said that you try to urge your students to reflect on their responsibility when they're doing this research. What do you say to them?
Yeah, so I teach like bachelor students and master level students in biology, where I lecture about like many aspects of biology, among others like the GMO. And then I say that to them that it's kind of much is here about opinions, but that they have like some kind of science-based opinion. So they have some responsibility to engage in this debate, so that we don't run into the issue that society is not informed, because I think that a lot of it is that you don't understand kind of the benefits. It's kind of this scary thing. The less you know, the more scary things are. So I think that, so that's also why I think that we as scientists also have a responsibility to engage in those debates. I would say that perhaps, at least I think to inform society, of course, to reflect and to get inputs. But I think that, say for the more microbiology discovery of CRISPR-Cas, I don't know how much the public would benefit from the dialogue with the public when discovering the
Well, it's an important point, because one of the amazing things about CRISPR is it's so sexy because of the speed also that you can do things. And the speed of science and everything, is infectious. It's exciting. You can do things cheap and you can do it fast and you can get some cool phenotypic results, really, especially if you're using model organisms, fast. Salmon is a non-model organism, so you get those things slower, but still they were getting it relatively fast. But the engagement part, a lot of scientists, they don't like it because we're actually, with RRI, we're slowing down the science. We're asking people to say, why are you doing what you're doing? Could you be using a different approach? Why do you use this method? And all these why, and deep reflection, which we would like to call it epistemological therapy, but some scientists are like, no, I got to get in the lab. I got to get that paper out. I got to do this and this and this. The speed of science now is very fast. So when we did that walkshop is when you walk hytte to hytte, cabin to cabin in Norway, it's on a 3 or 4 day trip as a way to slow down the science effectively. No Wi-Fi and no this and that, and we walk and we talk, and then when we get to the cabin, if there's a shower, we take a shower, but then we have an evening session like this where we go in. Sometimes walkshops have a lot of saltwater tears associated with them because there are a lot of issues in the lab that are not resolved, not talked about, a lot of ethical concerns that are built up, perhaps particularly with PhDs and postdocs, that there's never any outlet to really say, you know what, I don't like that my fish had 2 heads. This is a really big, again and again, it keeps having 2 heads, and I don't think we should be doing this experiment all the time. But you can't say that when you're really working on that important Nature paper, and you really the lab needs that to get funding. So we try to slow down the science. And another activity we did with our sterile salmon project is I took the scientists to the Bergen Art Museum, a museum where there's some modern art, abstract art. And we first spent 90 minutes just wandering, and they had to pick their favorite work of art that they could kind of see a vision of CRISPR in. If there was we had talked a lot of philosophical things about CRISPR, but can you find your favorite work of art and then conceptualize CRISPR within that work of art? And Anna Vargelis was sitting there. She sat there for such a long time, and she said she used that piece of art to really do some deep critical thinking about, and she had a most beautiful recollection that it was really, really cool, and she was just so grateful for that we were able to slow down the science so she could actually reflect. So it was a very positive experience, and the fact that we continue to work and have RRI on all of the salmon projects for 12 years now, I think is a reflection that the scientists are benefiting from it.
Mm. Yeah, I was there, so I kind of, I think it definitely had an impact on me just being part of the workshop. So yeah, but I'll come back to the workshop. But there's another question I would like to ask Professor Šikšnys as well. And do you think that, you're coming from your area of Europe where you're going to be doing your research, do you think that there's a difference in the role of ethical and moral issues in different areas of the world, different areas of Europe? Do you feel like you encounter different questions related to CRISPR in your area of Europe versus other areas you go to?
No, not at all. I don't feel any difference. The questions that are asked in Lithuania are exactly the same questions that you probably ask here in Norway. So that's kind of, there is no big difference. So yeah. But of course, people are always worried, and this is kind of understandable because you have new technology, and new things always scare people because they do not know how to behave with that. But if you look backwards, so actually I'm old enough to remember the revolution that happened before the recombinant DNA revolution. So that happened in 70s. And that actually then people got tools that enabled to manipulate DNA and then transfer DNA from one microorganism into another. And then actually the questions that were raised were exactly the same, because there were questions that scientists will engineer microorganisms that kill the world and so something like that. So because the technology was new and people was dealing with new things, so there were always what will happen next. But somehow these issues were resolved. And I think that actually with CRISPR we'll also come to the same solution that actually will be kind of resolved and will be accepted by public.
So, I mean, yeah, Trygve.
No, but I agree on that, and I think that public acceptance for technology is very much based on showing what it can be used for, that is meaningful for them. And this recombinant DNA technology was even bigger, I would say, the global reaction to it, because it was completely new to clone and manipulate. But then relatively rapidly, you could produce human insulin in bacteria, recombinant, and you could make better insulin and cheaper insulin. And then it was something that you could see the benefit of, which I think was important for the acceptance of the technology. And you mentioned also GMO. Everything came out wrong, I think, in the start of this. Big companies, spread of antibiotic resistance genes, and so on. And then also on top of that, in Western countries at least, we don't need it. Like GMO food, I think we don't need it, because and then the balance between the benefit and the risk is completely out of balance. So then it's easy to say that this is something that we don't want. But I think that this can change in the future, in particular when it comes to food production. And then with the CRISPR, we can also, there's two things. We can get rid of antibiotic resistance problem. We can make it safer. We can have more genetic control on what we do. And then if this can solve problems related to the food sector, then I think that this will have an impact on public acceptance also for the technology. We saw it with the vaccines under COVID. RNA vaccines. I think it would have taken a long time for them to be accepted, used in humans, if it wasn't because of COVID. But then we had a crisis, and this was the solution, and rapidly we started to use it. So
you're saying that we're all genetically modified now? Is that what you say?
yes, temporarily.
Temporarily, that's good to know. Yeah.
Yeah, so I would like to continue what Trygve said. So actually the acceptance of GMO is different, because the example that you provided, that insulin made by recombinant microorganisms, so it's made by organisms that by definition are GMO. Yep. But public accepts that, and actually drugs made by genetically modified organisms are okay. But when you start thinking about food or some other things, no. So then there is a rejection. So that's kind of also...
Yeah, but the crisis was right here.
Yeah,
right? Obviously, I can't leave my house if I can't. So it's a very important point, I think. And I think, when all of us in here are scientists in one way or another. And if we look at, for example, in political science in Bergen, we had a famous professor, Frank Aarebrot, very famous for Norway, political professor. And who did he want to study? He studied democracy by understanding who are the people that specifically choose not to vote. Because that, for him, as a political science researcher, was interesting, to understand democracy by people who opt out when you have a democracy, but you're not using it. For vaccine and COVID, it's very interesting to understand who are the people that consciously made the decision not to be vaccinated. Let's study those people, because I think we would get new views on the science and what public acceptance is and what it is not, understanding, the whatever type of views that these people had. That's very, very rich information that's very useful to understand and reflect about the technology. So I think it's, the majority of Norwegians did, but there are many that didn't. And that's that would be a very interesting RRI research question to go in and say, so how do those people how do they make the decisions? How do I make decisions on behalf of my family on what food I buy? We have a big debate now in Norway and in the UK and other places in Europe about ultra-processed foods. This is a technology, industrial food production, where they've been putting in all these chemicals into our foods that make it really quite unnatural and have been shown in systematic reviews to have an effect on cancer risk. That doesn't come into the dietary recommendations. They want more clinical trials, but the point is, when we see that there could be a risk of big, either big pharma or big food or big sugar or all of these big corporations, messing with our daily lives, essentially. You can't go into a supermarket today in Norway without 60% of the food at Rema 1000 is considered ultra-processed. And that has just gone totally under the radar. So it's like understanding why people make decisions, but they make decisions on the information they have, and it gets into engagement. It's a very, very rich research area.
And we won't go into the rabbit hole of the whole vaccine debates, but it gives me at least a little bit of leverage to go into something that's relatively adjacent to that. And I think that most of the arguments that I've heard about the importance of doing CRISPR research is about solving really urgent problems. It's always a sense of urgency in a way, and validly so. But since I am a philosopher of technology by trade, I love to just kind of dwell into the let's call them instrumental aspects of these questions, which is kind of going into the philosophical deep end, but just kind of bear with me here. Because I remember very distinctly on the workshop we did, there is a Norwegian very famous, at least in Norway, philosopher called Arne Næss. He was the founder of deep ecology, which is kind of, let's basically say that he had a theory or a thought about the intrinsic value of all sentient beings. I think even also plants, I think, but all living beings, I think. And he had made quite an impact, I think, in North America and Eurasia. Not so much in Norway, a little bit, but yeah. But at least I think that he's quite famous, of course. But we went to his cabin. That was the point. The workshop.
It wasn't a point. That was luck.
It was luck.
That was luck.
it was at least an interesting part of it.
A very interesting dimension of the workshop.
Because he's been really dabbling with these deep questions of ecology and sustainability and how we can kind of live in harmony with nature. And then I was finding myself sitting on the cabin wall of Arne Næss's cabin. It's a famous cabin in Norway. We were big with our cabins, by the way. And I was thinking, we're having a debate about CRISPR in salmon, and they're talking about their concrete examples of how this looks. They change color, then they're sterile. And I was thinking, the one question I had when I was sitting there listening to this talk was, what would Arne say? And that question just stayed with me, and I haven't really been able to answer that. And I was just wondering if any of you, I mean, of course, you're free of this question, but all the rest of us, what would you think would Arne say about this? And I look at Marianne. I'm sorry to put you in the hot seat, but yeah. Are you familiar with this thought? You could kind of opt out and say I'm not familiar with it. Yeah.
I would perhaps guess that he would be reluctant.
Yeah.
And that could be because, of course, he loved to stay at Tvergastein. He never he never fired so that it was 10 to 12 degrees inside the hut. He just wore his woolen jacket. So we could do that. That could be kind of a strategy, not kind of taking these advances that we have made and just go back to nature and then, So it could be a strategy. I don't think that it's the best strategy.
You're saying basically he was moral by avoidance, basically, or?
Yeah, he was lived a quite ascetic life.
That's true.
No, it's not a criticism of that, and he was also very tolerant to others. But I would guess that he would be reluctant, but perhaps since it could be of good to poorer parts of the world, with the food sustainability, that could be, you could see that as an advantage. But for us who don't need it, for instance, why should you mess with the nature if you don't need it? I don't know, but yeah.
Yeah, no, with regards to Arne Næss, I completely agree. That's all. It's difficult to say, but he was a little bit ecology philosopher or something like that. So I think that. But but back a little bit to that about what can it be used for, and I think when the Ukraine war started, we for a short moment had a potential food crisis that could even affect Norway. We had. And that was the first time I ever figured, is it possible? Can we have a food crisis in Norway? But then I thought, yes, we can in principle. And this is not yet over. And then you have the climate change, where big areas cannot be used for cultivation. So there are things going on that can affect how the global production and distribution of something as simple as food for us can be affected. And then I think GMO could be one contribution to this, not the solution, but one contribution. We can make species that can grow under different climates. We can defend insect damage, virus diseases. We can have more efficient use of the ground, more dense plants, and so on. And I think that very often it comes up that, no, we shouldn't do this, we should make sure that it was a better distribution in the world. And I don't disagree, but that is also difficult alone. So I think that we need many different approaches to meet the crisis that potentially can come. Maybe not far away in the future, but that can come. And there I think that GMO could be one such approach. Yes.
All right. You have
I think also like we should remember that evolution has everything that we see is run by evolution. We have had breeding programs so that we have the domesticated animals that we have the crops. This is kind of an efficient means to just speed up evolution, very precise and targeted. So it's what nature does. It just does it like millions of years. Now we could just do it next year.
Only we do it.
Yeah.
But okay, so now we're into the deep end, of course, and we're now ending our discussion very soon. But then my question then as a philosopher would be to say, okay, it'll be maybe a speeding up of evolution, and now we can speculate, I would argue, that isn't it so that it provides or it gives us or grants us an enormous moral responsibility because we're going in and we redefine our role as shepherds of the genetic legacy of a lot of different animals, maybe, and potentially ourselves. So what does that tell us about our own role in the future of our genetic makeup in a way? It's relatively overwhelming to think about, if you ask me. But have you thought about that at all, this irreversible aspect of this? Because it is irreversible, right? Is it? When you first go in?
Well,
yeah, so I can just comment on that. If you look at plants, so most of the plants that we are using for food, they are mutants. So they are natural mutants that occur in nature. Or were breeded by scientists. Yeah. And actually, if you look at regulation of plant genetic engineering, so you can find that actually you can create mutations by irradiating plants by gamma radiation. And this gamma radiation creates a lot of mutations. And we are okay with that, saying, oh, okay, it's safe and we eat that, no problem. But if you take CRISPR and introduce a single mutation, then it's not allowed. So that's the question, why? Why it's the difference? And then actually, if you make a mutation, if mutation happens in nature, or I make mutation using CRISPR, using in these mutations, there is no difference. There is no scientific method that allows you to discriminate between these two changes. So why one change is accepted, another one is not? So that's probably the question that is difficult for me to answer as a scientist. Yeah, so that maybe there are other questions, but yeah.
Sorry.
I just want to comment. I think these are very important reflections, right? So what makes something acceptable? Modern agriculture, or the last 1,000 years of agriculture, we've done more to genomes than we are doing now in CRISPR. With SDN1, for example. But I think a very important point here to be made is science doesn't do itself, people do science. And through this type of reflection, personal reflection, I'll give you an example of a really scary comment by Craig Venter, who one of the synthetic biologists in California, the Craig Venter Institute. He was asked by a journalist on a national news in the US, are you playing God? Mm-hmm. And he looked at the journalist right in the eye and he said, I am not playing. That's like blinking, warning, warning. What are these guys? What's this guy's motivation? What is your motivation here? Is it to, crack some really fascinating basic science questions, like really get understanding the genome for good, or are you really trying to get that extra profit, trying to build a big institute to fund? Very skeptical things. The Bill and Melinda Gates Foundation really supporting CRISPR for drought-resistant crops in Africa. There's great results in Kenya using CRISPR on bananas and things like this for basic human nutrition needs. This is kind of the easy, but that same technology in the wrong hands, it's the Collinridge dilemma also. It's like once you open up that Pandora's box, you can't go back. And this reflection that we need scientists to have this type of ethical reflection, I would say, this really happens in kindergarten, first grade, second grade. Like, what am I doing? Why am I doing this? Am I hurting or am I helping? Or what are my motives? This should be part of human nature, not just for scientists, but for everyone, because it gets people prepared to participate in the debate.
And on that note, though, even though I really want to delve into the human enhancement part of this discussion, I think that instead, since we're a little bit short on time, I think we just have to open up if there's any questions in the audience for the group. There's at least one. I think there's several there as well. We'll come to you later, or even three. Oh my God. Yeah.
Is on?
Thank you so much for this really interesting conversation. I'll just cut to the chase. And I know you talked about we as scientists should be engaging in these conversations and reflecting on these issues that you've talked about here. I wonder if you have ideas or you've explored ways in how you can do this in your daily research, in the context of at what point do you think you want to talk to people outside your science, outside your team? What point do you think it's important now? I need to speak to other people and get their input into this, get their perspective.
Any of you want to answer that? Marianne, maybe, or?
Yeah, I don't think that if I heard the question completely,
but public engagement in the scientific process itself, yeah. And
within the scientific process.
At what point
when to engage?
Yeah, I'm a bit like we talked about PhD students. I must say that they have extremely much on their hands to get this degree, to get this publication. So that's also why to slow down this research is actually quite challenging for them. Like in the experimental world, they have in total 2.5 years to do their PhD. If they come from another field, is almost not a chance. But we could do it like in the credits the mandatory credits that they do have. That could be like, you could have discussions. But say like panel debates like this, the students themselves could organize. They are extremely good at organizing, and they could be challenged to kind of have the roles of like pros and cons. So they could be challenged in that way. And they could also, if you look at the public, they are kind of, they will come. If you have debates like this, or not debates, but conversations like this, and invite the public, they will immediately come. They are very, very happy to attend such events. So I think it's about the challenge, but I think that if the PhD students should organize such things, it should be part of their mandatory credits and not take from their research time. So I don't support slowing down the research as such, because they don't have the time for it. We have the time for it because we sit there in our positions.
Well, the thing is, do we have the time to spare to not to do it. That's the reverse argument. But that's a debate. It's a very legitimate concern. If I may, Trygve, you had your hand up. I think that the thing that I think is super fun is bringing these discussions to the dinner table. Having just like practicing, like this is what I'm doing in the lab, and practicing explaining what you're doing to people who aren't in the lab, and getting feedback, like how do I simplify it, and where go, and getting new ideas, and then being just talking at Christmas time and family occasions, and like what you're doing to kind of get up that confidence that I can go out into the public event and talk and be understood. I think that's a really good start. But I totally support that. I mean, student organizations bringing this up in these type of evening events. But one way that we have done it in research proposals, so for all of you who are writing research proposals, to keep yourselves in business, putting in the communication or an extra work package where you have an RRI philosopher coming in. What I do is that I have monthly colloquia with my PhD students with an open agenda to reflect on their science. And they love it. And actually every single person who's been in this colloquia is writing an extra chapter on RRI dimensions as part of their PhD. So that's a great result because we're creating time and space in their normal schedule to reflect, and that is supported by the Research Council, very much supported by the Research Council, to have this built in to projects. It makes you more competitive, and it makes you better science. So we're not asking, RRI shouldn't be the cherry on top, and it should be really integrated throughout the whole track.
Yeah, and I think just a very small, I think that's a great idea, because then you also have a safe environment to discuss and reflect. Like for most PhD students to sit here like with this audience, they wouldn't feel very comfortable to discuss those.
No.
No, I was just thinking that, I mean, if you want to have funding for biotechnology research today, you have to answer a lot of questions about sustainability, bioeconomy, circular economy, things that are important for the future. And I feel that when I think about the master's students we have and also the PhD, they really feel that the research they are doing is important for all the good reasons. So that's what I think. And when we also try to map we have an increased recruitment now of people who want to study biotechnology. And then we can try to find out what is it? Is it because they get very good paid jobs in the end? No, it's because they have a high interest in the big challenges that biotechnology can be one solution to. So that's their motivation. That is what I feel is the everyday life.
We have time for a few quick questions more. I think at least two. Yeah, there you go.
Hi. So this has been a really lovely discussion. Thank you. And what's, I think, really beautiful that has emerged from your discussion today is this discussion of time, right? The time to do research, the time it takes to do research, how much time do we have to reflect or not, and can we or not afford to do this? How to slow down science, like you talked about with your walk. What was it called again? The walkathons?
Workshop, not a workshop,
yes,
but a walk.
Yeah, the workshops. And also very lovely. And then it kind of comes to that comment that you made that, and many of you commented on, that what we're doing here is just accelerating evolution, and that is a dimension of time. And it reminded me of a discussion we once had at a dinner table with guests who were non-scientists, amongst them philosophers. And so we talked about, well, what we're doing is exactly that argument. We're just accelerating what would have happened anyway. And someone asked a very good question, to which I don't have an answer and would like to hear your opinion. And the question was, yes, you can do that for that item, you have accelerated time. But you've lost all the opportunities of the environment to influence that evolution. That hasn't happened. So is it actually the same thing as evolution? Would you say that? And I thought that was a good question. I made an answer, but I'd like to hear yours.
It's profound. It's exactly the fact. You're playing God. You've chosen what you want to accelerate by holding everything else still.
But that's what you do with the normal breeding programs, with everything. We are in the age of the
but at the normal breeding, yes, in principle, but not necessarily the speed, the temporal component. I would argue is
what difference does that make?
Well, that's that's the
Well, I don't think
essence of the question.
I agree with evolution, like in nature, where you have kind of then there will be animals and plants that adapt to the environment. It will be fast. It will be quick changes. But that's not what has happened with the agriculture, for instance, for the last hundreds of years. We have kind of played God, and we have manipulated with these crops, but not with the kind of with the meadows or the like in the same way. So it's kind of we speed up something, but we don't take care of all of the rest of the ecosystem.
Well, there's something called cultural landscape. So we have decided that this is part of who we are, humans living in the environment, man and nature. So yeah, there's a certain amount of things that becomes culturally acceptable. But I mean, CRISPR is similar, of course, to modern breeding, but it is unique in the same way. So we can't really forget that. And I think that, you know, at the dinner table, it's beautiful when these questions emerge, because it really is, we really see it from a different perspective. And we as scientists get conditioned to, oh yeah, but this is just how it is. This is how we do it. This is how we do it. But the question is, why and what is changing? It's an amazing vision to see, you know, tens of thousands of evolution in an experiment that maybe lasts 3 months. So I totally support dinner conversations, because it gets us into some deep philosophical thoughts.
I agree, but if you think about animal welfare, so if you do a normal breeding program, you'll kill all those cows that didn't produce that 40 liters of milk that we have, this Norwegian cow production that we have today.
Yeah.
So, but then if it was possible with the CRISPR to go tomorrow and say that, okay, we know that 40 liters is kind of sustainable level, we could save all those thousands of cows, like just to, yeah.
Yeah, of course. It's a totally legitimate argument, right? And so it's at the end of the day, you know, it's in democracies, politicians kind of have to decide what is prioritized, what uses, and what is not, what is risky and what is not. But those discussions all these arguments have to kind of come out in order for us to say, okay, there's no perfect, CRISPR is not a silver bullet by any means, but we have to make some decisions as a society on what we prefer.
Probably this discussion is going to continue after the session. So, but just to have time for a few more questions. Yes, very quick.
Yeah. Okay. My question is from 3 perspectives. 1, nature, like the sun, emitting radiation and causing natural mutation that affects nature and sometimes result in something different, which is similar to what science research is doing. The other one, research being too fast, and we really don't want to open the Pandora box because we don't know what may happen. And then the opportunity cost of taking that part of action. The third one is the public having a say in what is going on, which we know is influenced by their knowledge level, because technical details, not really everyone in the public have in-depth knowledge. So how do you really contribute to what you don't know? The first one, could it be that due to this natural mutation is the result of some of unexplainable sickness, illness, and condition we cannot really explain in research, because nature in itself sometimes is causing nightmare to natural blessings we're supposed to have? And then another part on our research is we really don't need science to convert nature to nightmare. For instance, the fire, the water, these are natural phenomena, but if we understand them, we can use the fire. But sometimes even the understanding of fire can enable us put up fire when the fire, we don't want it. But if we say, no, fire is dangerous. I don't want fire, please. And then there is fire, but we need to put it up, but we don't have sufficient data. We don't have sufficient knowledge of what is going on. We will have destruction. So I'm afraid, because this is an emerging field, and a lot is happening. For instance, I'm not in medical science. I'm more into AI cybersecurity. But one thing I've known from my own research line is that no matter the number of use cases you try to experiment with, there are several abuse cases, misuse cases you cannot think of. And sometimes you have to let it go in a controlled environment, which in our security we call it sandboxing, and let it misbehave, but it's within your control, and then you see the behavior, the pattern. So I'm afraid that with this concept, like, let's reflect. Let's not open the Pandora box. Are we actually doing good, or are we doing harm? Because if we don't have sufficient data to understand the problem, how can we even call it a problem in the first place? If we don't know enough, we need like flood of knowledge, flood of light to see this from every dimension, perspective possible, because definitely we know something, no doubt, but we don't know all things. We're even making criticism sometimes on things we really don't understand. And from my perspective, the way we can understand this is to do more research and be in control, not like it's happening randomly. We set the condition, the criteria, but that cannot happen from a PhD candidate perspective. But from someone like a panelist, you are respected, you are known. And the public we are looking to, they have some knowledge, but they don't have details like us. They know more that they have little. And sometimes what they know is the opinion of just one person who is in the TV show or in the newspaper, and it's an opinion, maybe based on incomplete facts. So what I'm saying is these three perspectives, can you people like throw some light on them? The public opinion, are they based on facts? Are they based on sufficient knowledge? The controlling research, is that the right thing?
I think maybe some of you can kind of step forth and answer some of the questions there, because there were several. Incomplete knowledge, scientific uncertainty, Pandora's box, and the responsibility of researchers. Anybody have any comments on that?
Well, I think there were a lot of very good reflections there, and one of them that I caught was, how do we talk to the public if they really don't have can't really just grasp basic science to begin with, because of maybe they never been to school before. There is not a public school system, or all these other systemic issues. I think it reminds me of the debate that a local community of mine had on gene drives. So gene drives go like way beyond just simple CRISPR, and it was an effort, a company that came into an island community, Key West, highly educated Western island in North Caribbean, because of the West Nile virus, and they wanted to use gene drives to make sure that, no, it was Zika, I think it was Zika, so that mosquitoes couldn't spread it. And the community was scared. They're like, we were not sure if this is a good idea, but then all the industry was bringing in all the people say, this is safe, we've tested this in Brazil, trust us, and this is gonna be a public health intervention that is going to help all the pregnant women so they're not going to get Zika, and all these promises. But then I happened to be there, and some of the community members asked me to talk about CRISPR. They said, we have a feeling that there is some risks that are not being communicated, but we don't know what those are. Could we have a discussion? And we watched one of these CRISPR documentaries, because I think that watching the films and the community of journalists that are used to taking complex situations and putting that into a narrative, so it's easier for people to kind of grasp what's going on in a more storytelling way. We watched that, and then we discussed for the next 2 hours about their local issues and what they really care about, what are their universal values, what do they really want for their community. And ultimately, the community said, no, we do not want this. We do not want to open the Pandora's box. And I think it was a very smart idea, because there's not a Zika problem in the islands. It was a problem that was perceived as being bigger than it actually was. And so I think that this is going back to the arts. This is why we have wonderful colleagues at the university who can help us people who are even illiterate can understand storytelling. That's how we've passed information through, hundreds of thousands of years of human civilization. So we can get back to basics and use art and use different modes to understand, so people can understand, is this something for our community or not?
I think that there's at least several other questions that was also proposed, but I think we just have to cut to the last question. We're way over time. It's an engaging topic. I'm glad. Last question before we end.
Okay. Okay, thank you. I really enjoyed this discussion. I want to raise one question that I asked myself in the context when we put GMO and the GMO flop in context with CRISPR. So you attributed a lot of the things that were not working with the introduction of GMOs into society to a lack of RRI. But I want to ask the question, is that maybe also to be attributed to the exploitation of intellectual property rights in that context by the actors that have been back then active there? And then my question is actually, would you see that a similar danger for the use of CRISPR in society right now, given the current intellectual property rights situation with this technology and the patenting?
So the intellectual property is a very different world. So that's kind of very different from what we're doing in the lab. But anyway, so of course, there is still ongoing debate who will own the CRISPR technology. So that's kind of this Broad versus Berkeley or Berkeley versus Broad debate that it's still ongoing. So currently, the kind of the rights are with Broad, but as far as I know, but then Berkeley actually claimed that actually probably it's not right, and that it's still ongoing. And of course, this to some extent actually hinders the research also and development, let's say, of small companies, because if it's not a clear issue who owns the rights for CRISPR, so then for small companies it's really difficult to get, for example, license both from Broad and Berkeley. So they cannot simply afford that. So that's kind of, but it's kind of the legislation framework that we have to live with. So that's kind of,
Yeah. And I have to mention, I just had to say it, that since I do my PhD in biotechnology patents, this question gives me like a rash.
Who owns?
Thank you for bringing it up. It's at the end. That's great. I'm going to scratch my way to sleep. I would just say to the panelists, thank you so much for a very insightful and broad discussion. Thank you for coming. Thanks.